Good evening. I think you can hear me right properly with this new microphone. Okay, good evening again. My name is Eva Muñoz, and I'm going to guide you through the last part of this workshop, WIDEN-ITC. And first of all, I would like to mention a slight problem we are having with the Wi-Fi connection here in the room. So at this moment, we have limited connection, so unfortunately, some of you might not be able to follow me with your computers in the AFFINimeter tutorial. But for those of you who have your own Wi-Fi connection, for instance, EduRAM from the university, I think you will have no problem. Even you can share your computer with other colleagues, so you can, I think we will make it work, okay? So in this last part of the workshop, I'm going to talk about isothermal titration calorimetry analysis. This is a tutorial, and it refers to ITC analysis with the software AFFINimeter. In fact, it is not just one tutorial, but 3 tutorials where we pretend to describe the main functionalities of the software AFFINimeter. I will talk in the first tutorial on how to create your own binding models using AFFINimeter. A second tutorial talking about how to simulate ITC isotherms. And finally, I will talk about how to analyze ITC data using the advanced fitting tools of AFFINimeter. These tutorials are going to be interactive, so whoever wants to follow me performing the examples I'm going to show, so you are welcome to try them yourself simultaneously. And remember that you have the live chat room too for those following us online, so you can ask questions anytime, okay? Well, before starting with the first tutorial, I would like to give you a brief idea on how AFFINimeter works. This is the toolbar of AFFINimeter, and I wanted to point that we have 2 main parts. One part that we call My Lab, where we basically store datasets. Datasets regarding all the experimental data we need in order to perform the ITC experiment. And we are going to organize these data sets in different parts. We can store the specifications of our ITC instruments, also the ITC data series, experimental data, and also we have also a list of binding models that we can use to fit this data. Additionally, we have this part that we call the project management. That is where we perform the simulations and the fitting of the ITC data. So what we have to do with AFFINimeter is to prepare all the experimental information that we need, organize it in these packages, and then we have just to combine the information we need, the ITC experiment, the ITC instrument, experimental conditions within the data series, binding model, and we combine it together to generate our project into the project management to perform a fitting or a simulation. So this is basically how AFFINimeter works. And from this general idea, I think we can start with the tutorial 1. Create your own binding models using AFFINimeter. So let's go to the AFFINimeter software now. So this is the main like login window of AFFINimeter. So just log in into your AFFINimeter account. Click on login. Double click in my case. I sometimes forget. And okay. So this is what we see when we just enter into the AFFINimeter account. And in order to design, in this first tutorial, I'm going to show you how you can design your own binding models using AFFINimeter. And for that, we have to go to My Lab in the menu bar, and from the drop-down menu, yes. Yeah, because in your AFFINimeter account is the information you have at this moment. I did some more experiments in my account. So yeah, this is just a visualization of the last projects or fittings you were working with. So we may have different things, okay? So yeah, from My Lab, go to the option Models, double click, and what we have here is a list of binding models that we have in our account, and in red you will have a list of basic models already implemented in AFFINimeter. These other models, I created them myself. So let's have a look at how a binding model looks in AFFINimeter. Just go to the action column and click on this button where it says View Model. I'm going to click on the model, the simplest binding model, 1-to-1. Okay. Yeah, I'm used to just one click with my computer, and this needs double click. So this is how a simple 1-to-1 binding model looks like in AFFINimeter. And as you can see, it's pretty simple and straightforward to visualize, in my opinion. We have the free species consisting in two compounds, species M and species or compound A, that are going to interact to form a 1-to-1 complex MA. So this is the simplest case where we have one single equilibrium, but we can define complex binding models with more equilibrium, with more equilibria, a combination of equilibria. So let's close this window, and let's have a look at a more complex model. Let's go and visualize the competitive model. Okay. So in this case, we have again the free species. We have three species, compound M, compound A, and compound B. And in this situation, what we have is the formation of complexes MA and MB. And this means that that compound B and A compete with each other for the establishment of a complex with M. So this is an example of a binding model with two equilibria. So from here, how can we design our own binding models with AFFINimeter? Well, we just have to click in this button up on the right, Add Model, and immediately what we see is a window that pops. That is the reaction, what we call the reaction builder. In the reaction builder, we are going to be able to design one equilibrium from that will form part of our binding model. As you can see here, we have a reaction scheme that is empty and that we are going to use to design this equilibrium. Here we have the species. Sorry.
I'm not able to put the name of the model, so I'm not being able to follow you.
Yeah, if you want to open the reaction builder, you have a button on top.
Yeah, I clicked on the new model.
Yeah.
Then it's walking in the model name, and before I give it the name, it doesn't allow me to follow your presentation.
No, once you click on the Add Model button, I did nothing else. Just this window should appear. Okay. If you need me to go slower, just let me know, okay? So yeah, I was saying that we have the reaction scheme that is empty, and we have here the species that we can use in order to design our equilibrium. We have the compound or the species M that stands for the main compound, the compound that we have in the calorimetric cell. A stands for the compound, the main compound, which is in the syringe, the titrant, and B is a third species, for instance, a competitor that can be located in the cell and or in the syringe. So in order to design our equilibrium, we just need to select, click on the species we are interested in, drag and drop into the reactive box. And as you can see, immediately the something appears in the new box that corresponds to the complex form. I'm going to drag and drop compound A, and right now what we have is an equilibrium corresponding to a 1-to-1 binding, the same we saw previously. At this point, we can modify the stoichiometry of the complex with just changing the number here next to M or A. For instance, I can generate a complex with 1 molecule of M and 3 of A. Even I can just eliminate compound M and consider an equilibrium of oligomerization. An interesting thing of AFFINimeter is that we can generate binding models of dissociation process, but or oligomerization process, not only through the formation of a dimer. We can increase the number of monomers. So this is a new binding model that we can create that is not available in other ITC softwares. So I'm going to go back to the 1-to-1 binding. I'm going to get M again. Just as an example, even we could consider the formation of this type of complex, MAB. I'm going to eliminate B, and I'm happy with this equilibrium so far, so I will click on accept. Now we have our equilibrium, and if we decide to finish with the binding model, we just need to name and save the model, but I will go a little bit further. I'm going to add more equilibria. And in order to do that, I could choose to add more equilibria or one more equilibrium starting from the free species or from the complex form, in this case MA. In order to create a new equilibrium from the free species, just click on free species, and this list of arrows will appear. Just select the orientation you prefer, this one. And again, we have the reaction builder to design the next equilibrium. Again, we have the species available here. We have again M, A, and B because we are starting from the free species. And I will just repeat the sequence by dragging and drop the species M and the species B. I will click on accept, and now what I have just done is to create a binding model that is the competitive binding model that you saw a few minutes ago. We can edit or delete equilibria at any time. Just click on the asterisk here next to the arrow of the equilibrium, and I'm going to delete this one. Okay? We could edit it. If we edit it, the reaction builder will appear again, and we can do modifications in this equilibrium. So I'm going to delete it. Okay, accept. And now I'm going to add a new equilibrium starting from the complex MA. Select the arrow you prefer, and we have again the reaction builder. this time the appearance is a little bit different because we are starting from a complex. So we have the initial complex MA, and we have one more species available here, that is the complex MA. I could consider, for instance, that the complex MA is forming a dimer, right? So I think this is very useful because it gives you a lot of flexibility when the time comes to design binding models based on realistic situations. So I'm going to eliminate this one, and what I'm going to do is to add a new molecule of A. Click on accept, and I will add a new equilibrium to the species starting from the complex species MA2. Select the arrow, and I decided to add. Yes.
What is the meaning of the orientation of the arrow?
It's just well, it's for fun, and if you have many equilibria, you might want to order the equilibria somehow in a way that you see it, you have a better view of what's going on. So that's the only reason. So now we have complex MA3. Click on accept. Well, before clicking on accept, I would like to give you a little bit of information of this box, other products box, that you can use in order to consider that you have side products in your reaction scheme. For instance, if I consider I have finally a mixture of complexes here, for instance, MA, and when I drag and drop this species, immediately the reaction rearrange this new, the complex is modified according to a mass balance. Okay, I will eliminate this and click on accept. So this is in fact the model I was interested in designing. It's a sequential binding model where we are generating a final complex MA3 stepwise, and we have intermediate complexes. So from this binding model, we are going to get information on thermodynamic information on every equilibria described in the binding model. So I'm happy with this binding model, and I'm going just to give a name and save it. For instance, save model, and then the new model should appear in your list. Anytime you can visualize it with this button, you can copy it with this button, or you can edit it with this button here. Okay. Okay, so this is basically all I wanted to show you about how to design binding models with the reaction builder. And I may show you an example before finishing. This example that I call 1-to-1 plus dimer that I made myself. Just to show you how we can design very easily binding models that correspond to realistic situations. For instance, we have a 1-to-1 binding interaction where one of the compounds is forming a dimer. We don't have this type of binding models in other commercial software, easy to use. And you can see that very easily we can design complex binding model with multiple equilibria, but also, like, sometimes you just need 2 equilibria to define a system, a realistic system that's, well, sometimes it's not easy to solve because we lack of the appropriate binding model. So as I was saying, this would be the end of this first tutorial. Yes?
This assumes that the binding sites are fixed, right, and finite. We have a well-defined number of binding sites.
Yes.
Okay, so and the software is not flexible in that respect.
Yeah, well, there are some, there is a lack of flexibility to make, to avoid people make things that are not realistic, in fact, or...
This is only useful to binding to proteins. For example, binding to membrane is outside scope of the software.
Well, with the model builder, with the reaction builder, yes, we have to, it's useful for many applications. We have a second way to design binding models. I will talk about it in the next, in the simulation too, but very briefly, that contemplates the design of models based on independent sites. And you were talking, sorry, about
Membranes, for example, there are no binding sites.
yeah.
And the interaction with the DNA, for example, there are also not well-defined binding sites.
Right.
Polymers, other than proteins.
Right. So in the case of DNA or polymers, I would go for independent sites based on my experience. Not always.
Sites are not well-defined, so
Right.
it doesn't make sense to apply it with the binding sites.
Right. Right.
That makes sense.
Yeah.
The best.
Yeah. In some cases, you could make an estimation of binding sites.
There are no binding sites. So in membranes, there are no binding sites.
Yeah, well, I was talking about polymers or DNA, where sometimes you can estimate. But yeah, we should study that concrete system you are talking about. I'm sure Angel will be able to develop a mathematical equation for that. I trust on him. Any other question before continuing with the next tutorial? Well, it's just some...
indication on the delta H sign is going to be the one for the dissociation, or it does not have that implication?
No.
So the opposite sign.
Well, it's just a matter on how to name the model. In the reaction builder, we have the equilibrium, the scheme, and the information you obtain later on depends on your experimental setup. If you have a dimer form and you perform a dilution experiment, you will get the enthalpy corresponding to the dissociation. But if you perform the other, like the formation of the dimer, you will get the enthalpy, the information corresponding to...
What I mean is, when one looks here, dimer dissociation model, I would expect the equation to be A2 to give A times A, okay? And so I was wondering, since you called it dimer dissociation, that means that your delta H will be for the opposite process.
No, it's... Oh.
Do you understand my question? No.
Yes, I think I understand. You mean like what is going to give you? What kind of information is going to give you?
Yes, so there is when you write the model builder always starts with the Fourier species. So the way you write the model, you always have to start with the Fourier species. But all of these are equilibrium reactions, so it doesn't matter. So it's a way of making definition. So, but
all the association constants in AFFINimeter are correspond to the creation of more complex species. All of them. If you want the dissociation constants, you have to make the opposite. Yes.
That was my question.
Yeah.
So they all correspond to
Exactly, exactly. And so also all the arrows are bidirectional, so all are equilibrium. That's the
Any other question? No? Okay, so let's move to the second tutorial.